understand how the homework is done, what the answers mean. So exam on Friday. Again, the same format as the first exam. We'll talk about that on Wednesday. I'll have last year's exam up in the copy center for you. We'll have help sessions on Thursday, one in the afternoon, one in the evening. Hopefully we won't have a telephone meltdown, computer meltdown, and so we'll be able to schedule those things.
What day is the last year's exam going out?
I don't know, probably on Wednesday. So, there you know that. We left off talking about recombination. And we went through different kinds of recombination. We're going to look specifically at general recombination. Remember, the definition of general recombination was recombination between two pieces of DNA with very large regions of homology. Large regions of homology typically means chromosome-to-chromosome recombination. Whether it's two viruses infecting a cell, whether it's bacterial recombination, which is not chromosome-to-chromosome, but still very large pieces, or meiotic recombination. The model that we're going to look at is the Holliday model. All basic models of recombination follow the Holliday model. He was the first one to make a model for how recombination occurs a long time ago, and it's still good, but it's been modified. So this is going to be a modified Holliday model. We're going to do this in such a way that we're looking at a general universal system, but we're going to do it focusing on meiotic recombination. So meiotic recombination involves getting the chromosomes together and pairing up the chromosomes in first meiotic prophase. And that's always been one of the mysteries in recombination. And to this day, actual details, the mechanisms of how chromosomes pair up is still a mystery. If you really think about what's going on, the DNA in the chromatids of the duplicated chromosome, that DNA is double-stranded. And so you really can't use any kind of a DNA base pairing mechanism to look at the chromosome. The only hint we have is looking in a nucleus. And what you see in a nucleus, chromosomes are in domains. So a pair of chromosomes there, a pair of chromosomes there, a pair of chromosomes there, a pair of chromosomes there. And so for as many chromosomes as an organism has, I've talked about looking at the bowl of spaghetti in the nucleus, and that's correct if you just look at it without labels. But we can now label chromosomes very accurately. And so looking at a nucleus, we understand that chromosomes are not randomly distributed in the nucleus, that chromosomes occupy specific domains. So that's part of solving the mystery of how chromosomes pair up. But we're going to ignore that, and we're going to assume that we have paired chromosomes. So we have a pair of chromosomes that are brought into conjunction. So at this point, we're going to switch over to bacterial recombination. Again, it's universal. universal, but I'm going to use one protein in the bacterial system as the model for what we're doing. So the first step is pairing. We're going to look at a variation of standard recombination. So we're going to look at what happens when we introduce a nick. Remember, a nick is one broken ester bond. So one strand is completely intact, and we're going to introduce a nick in the second strand. And then what we're going to do is strip off that strand, bring it down, and put it in the second duplex. So pairing, nick, and then transfer. The protein that does this, it's not exactly an enzyme, it's called REC-A in E. coli. But every organism has a gene that's homologous to the E. coli REC-A gene and makes a protein that's homologous to REC-A. REC-A really isn't an enzyme. It's doing two things. What it's doing is, first of all, protecting. So we have the same problem that we had during replication. This is single-stranded DNA, and it's very susceptible to just general degradation. So REC-A is going to coat the DNA, the single strand of DNA. And then, the more important part is search for homology. What that means, basically, is REC-A is going to run this section of the DNA across the homolog until it matches up, complementary, of course. And so search for homology means that REC-A is going to move that piece of DNA back and forth, one base at a time, until it finds the correct homologous region. Everybody see what's going on? Once that happens, then we're going to make a second nick there. And so I purposely made that nick in a different spot. One of the mysteries before the Holliday model not only was how two chromosomes line up, but people have been looking at recombination for decades, going way back to Thomas Hunt Morgan. And what they found is that recombination is extraordinarily accurate. So things are coming out perfect from crossing over. No duplications, no deletions, no mutations. And so people always wondered, how is it possible to get perfect recombination without mistakes? And so this sort of solves that by lining this up and packing that down. We're going to do the same thing here. We're going to bring this segment up. There. And so, and again, coating it with REC-A at the same time. And so we have this structure here called the chi structure. Chi is the Greek letter for X. And so this is going to set the stage for things that we're not going to do. You can look in your book. And so what we have is this DNA there. gonna have to be removed. And then we have a gap. We have a gap there. And so the gap is going to be fixed by DNA pole 1. And so this is going to work just like it does during replication. If we remove the next Okazaki fragment comes up before we remove the primer, it's going to leave a gap. DNA polymerase 3 is already gone. DNA pole 1 fills in the gap. And so DNA pole 1 is going to fill in the gap, and then DNA ligase is going to seal up all these nicks. We still have that chi structure there. Chi structure is still there. We're just fixing up the 2 template chromosomes. Notice the involvement of DNA pole 1 and DNA ligase. We use DNA pole 1 and DNA ligase in replication. We're now using it in recombination. That's pretty much the definition of pleiotropic gene. So if you mutate DNA ligase, you are going to affect replication, you are going to affect recombination, you're also going to affect DNA repair. So a mutation in a gene that has multiple phenotypic effects, that was the original definition of pleiotropic. And so we waited for the molecular section to see what pleiotropic really means at the molecular level. It's a gene whose protein product is used in different systems in a cell. Replication, recombination, we'll see DNA repair later on. Any questions on this? Yes?
So what is the exact function of DNA ligase?
The exact function is the same as it was in replication. With a phosphate and a hydroxyl group, it doesn't add anything, it just makes that last ester bond. It seals the nick. So all DNA ligase can do is seal a nick.
And what happens to an extra class?
It gets degraded, it gets thrown away. And so the size of that flap should be the size of that gap in reality. That's really good artists.
And does anything take away that gap?
Yes, enzymes, nucleases, absolutely. Yep. We have to trim that away. Any other questions? Yes? So, right there, that is that. And so it's called the chi structure of recombination. Anybody else? Yeah.
What exactly is the difference between the functions of DNA pole 1 and DNA ligase?
So DNA pole 1 can actually add nucleotides and fill in a gap that's missing DNA. But it can't seal up, it can't make that last connection. DNA ligase, if all the nucleotides are there, it can connect a phosphate to an OH and make that ester bond. Yes?
Would the single-stranded DNA on the bottom also have the rec A?
Yes.
Okay.
Now, well, not there.
No, but
one
the, yeah.
there. Yeah. Yes.
In replication, DNA pole 3 does the synthesis on the other strand. What's the difference when you use DNA pole 1? I think it's the same.
Well, so pole 1, so you don't need pole 1 ligase on the forward strand.
Yeah.
You're just flying around. On the backward strand, you're bringing up that Okazaki fragment and bumping into the next one. And so if you remove that primer after that gets there, you're going to have this gap here. And so pole one fills in the gap where the primary was, and ligase always has to seal up what was out in fragments, no matter what. Anybody else?
What specifically are nucleic acids?
A nuclease, just, there are tons of nucleases in the cell just looking for single-stranded nucleic acids, unescorted. Yes?
So, they like move the X on the bottom line and, seal up the gap in.
Well, so nucleases remove that. But O1 and ligase fixes the top part. You have a question?
No, I was just gonna ask like about the, what do you mean with that pleiotropic or pleiotropic gene?
Pleiotropic gene, multiple phenotypes. That was the definition that came up on section 1. Anybody else? Any last questions? Yeah.
Rec A just protects that single-stranded.
Well, no, Rec A does two things. So, Rec A protects, and probably the more important part for recombination itself is search for homology. Anybody else? Okay. So the last chapter,
So what is it exactly doing?
protects and search for homology. The last chapter for section 2 for next week's exam is the genetic code. Again, remember back to 1952. 1952, Hershey and Chase settled the issue. DNA is the genetic material. Before that time, people assumed that the part of the chromosome that was the gene was the protein, and the DNA was just a scaffold that it hung on. Hershey and Chase changed forever what is a gene. And so there were many questions. How is DNA faithfully replicated? We just finished that chapter. What is the coding mechanism? How does DNA with 4 nucleotides code for a protein that has 20 amino acids? That's what we're going to do next. And then when we finish this, we're going to look at the third major question: what is mutation? Mutations have been known since ancient times. They were called sports. So, in 1952, people had to start thinking about how information is encoded in the DNA. This was right after World War II, actually, when coding was such a major role in World War II, breaking enemy codes. And so it was on everybody's mind. But they had no idea what the code could possibly be until they knew what the structure of DNA was. And believe me, you don't want to look back in the early 1950s and see some models of DNA. It was just ugly stuff, making models without any data. Until Watson and Crick. 1953, we had the double helix. And now, the structure of the DNA, as Watson and Crick showed, provided a model for replication. The structure of DNA also provided a model for how information is encoded in the DNA. So now people can sit down and play with different coding mechanisms. They knew a whole bunch of things. They knew that the genotype controlled phenotype. They knew genes coded for proteins. That was the big one. Well, that was really a problem because of all the X-ray crystallography that came before 1953, people understood definite shapes. Coatings have very definite folding patterns. It's like going to IKEA and buying a box to make a table. You have to put together all those parts. People wondered if that had to be included in coding. And so it didn't take long for people to figure out that you could take a protein and denature it. That is, you could unfold it, and if you drop the temperature back, most proteins would fold back automatically. Some wouldn't. That's how you make a fried egg. You take the egg proteins, heat it up, denature it, and they do not reform their proper structure. But lots of proteins do. Enough proteins could be refolded in the test tube without any additional information. The folding was not going to be an issue to concern people for the code. All people have to worry about is nucleotides, 2 amino acids. All they have to worry about, and they now knew that the DNA was a linear string of nucleotides, and they knew the protein, the unfolded protein, is a linear string of amino acids, and the fact that most proteins could refold means they do not need to put folding instructions into the DNA. So obviously, a 1-to-1 ratio is not going to be sufficient. You can't have one nucleotide coding for one amino acid. Even a 2-to-1 is not going to be sufficient. You can't have 2 nucleotides coding for each amino acid because you only have 16 possible combinations. And so we have this notion of a 3-to-1 coding ratio. But that has a problem. A 3-to-1 coding ratio gives 64 possible combinations. We have 4 nucleotides, and we have 20 standard amino acids. So a 3-to-1 ratio, which is the least you can have, has way more combinations than is possible. But this 3-to-1 ratio, the 3, which is what it is, you know, now defines a codon. Any questions on this background, this sort of history? So let's look at the features. What is the genetic code? So first of all, the genetic code is non-overlapping. Take this off. So we can make just a random sequence. A random sequence of nucleotides. So that's the linear sequence from the Watson and Crick model of the double helix. So we can imagine that there are 2 different ways to read out a 3-nucleotide code. We could read out 1, 2, 3 for the first codon, 2, 3, 4 for the second codon, 6, 7, 8 for the third, in a non-overlapping fashion. So that's one model. They didn't know anything in 1953. That's one way that you could get a linear code of 3 nucleotide combinations. But there's an alternative model. 1, 2, 3 specifies codon 1, 2, 3, 4 specifies codon 2. 3, 4, 5 specifies codon 3 in a fully overlapping code. So these were the two models that they had. The reality is that this wasn't a problem for more than about 5 minutes. Can anybody see a quick solution for how one could determine whether it's overlapping or not overlapping without doing any experiments, knowing that people had amino acid sequences?
Try to figure out one sequence, it's just one amino acid, just just all A. And then
Oh, all A is the worst one to do, because then you don't have a reference point. No,
how about two amino?
yeah, but they can't do that. I'm saying the data was already there for them.
How many codons are produced for a 9 amino acid?
They can't do that. You're in the 21st century with all kinds of technologies that weren't available then. All they could do was go to a library and look up protein sequences. Remember, they had lots of protein sequences from all the X-ray crystallography of proteins that preceded Rosalind Frank's X-ray crystallography of the DNA. So just think data mining, not experimentation. Just go to a library, look up a few proteins. There are actually 2 different ways of doing it. Okay. Anybody think of a quick way to do it?
Figure out proportions, relative proportions of amino acids.
I don't know what you mean by proportions, but
Figure out which is the most commonly used amino acid and see how often that codon, which is the most often codon.
they don't have the ability to sequence DNA. You're living in the 21st century again. It's all, see, they don't have any DNA sequence. All they have is amino acid sequence.
Because if you did it one way, you'd have it fold onto itself in the wrong pattern.
Well, but you have the amino acids are good on what the protein is. The question is, what was the code that gave rise to the amino acid sequence that you see in the library from X-ray crystallography?
So did they know the amino acid sequence?
They knew the amino acid sequence of lots of proteins, mostly small ones, but that doesn't matter. And let me give you a hint. They also knew the amino acid sequence of mutant proteins.
If you use the overlapping one, there'd be too many amino acids by the end because it goes 1, 2, 3.
They have no yardstick to measure to get too many.
If you compare it with the mutant, does the mutation change just 1 amino acid or 2 or 3?
Very good. Let's say, for example, we change 1 nucleotide, 1 mutation. They were pretty sure that was it. What's the consequence of changing 1 nucleotide in the overlapping and the non-overlapping code? How many amino acids will change with 1 nucleotide here?
3.
For what? How many, if you change 1 nucleotide here, how many will change in the overlapping?
3.
And they have lots of hemoglobin mutants, red blood cell hemoglobin mutants. It's just a matter of looking it up and determining that each mutation changed a single amino acid for as many mutations as they could find. That's the easy one. I don't even want to go into the more complicated. So the code is non-overlapping. They knew that without doing any experiments. No internal punctuation. So let's get rid of this. No internal punctuation means that once you start reading, there are no commas in the code, no semicolons, no punctuation internal. We're going to talk about external punctuation later, but for now, once you start reading, you read every single nucleotide as part of a codon. Any questions on what I mean by that? Yes.
Actually, I have more question on the non-overlapping.
Okay?
How
And
do
the
they
one I
know-
erased.
Wait, no, I'm mistaken. How did they know that? How did they know about the one overlapping?
Because a single mutation, they didn't know anything about it. Every mutation they look like only changed one amino acid. The prediction is for an overlapping code, it would change 2 or 3 amino acids. And that was really clear. They didn't, but the fact that every single amino acid, every single mutation only changed one, that was a reasonable assumption. Anybody else?
On the no internal punctuation, is that after the sequence has been processed, so like there's no introns?
Yes. So we're not talking about introns. Introns are removed. We're talking about the expression. So forget about introns for this part. Introns are gone by the time we get to read out the code. Anybody else? The code is degenerate. So we have 64 codons coding for 20 amino acids. Degenerate means multiple codons per amino acid. And the multiplicity is 1 to 6. So there are a couple of amino acids that only have a single codon. There are a few that have 2 codons. There are some others that have 4. And some of the most common amino acids have 6 codons each. Now, what we have then, we have 61 coding codons. 3 non-coding that we will talk about later. So 61 codons codes for all 20 amino acids. Any questions on what degenerate means? Code is non-ambiguous. That's good. That means that the code that you're using today is the same as the code you used yesterday and will be the same as the code you're using tomorrow. The code does not change. You have a code dictionary in your textbook. No, you don't have to memorize. That code dictionary is nearly universal. We'll talk about that in a couple of minutes. But almost everything. Non-ambiguous means not changing. Any questions on what I mean by that?
Degenerate, is that kind of the same thing as saying redundancy?
Yes. Yep. So there are redundant codons, but there's a multiplicity of codons for some of the amino acids, but not all.
Is the redundancy of each codon for amino acid associated with the necessity of that codon or that
Associated with what?
Is it associated with the necessity of the amino acid and protein? Like
For the non-ambiguous, it's saying that for non-ambiguous, it's saying that a codon in one person is the exact same as someone else.
It's always going to be the same. You don't have a codon coding for different amino acids at different times. It means that there are no internal punctuation marks. That every nucleotide is part of a codon. But we're not talking about each. Every nucleotide is part of a codon. Anybody else? The code is co-linear with protein and the DNA. That means if you have a gene and you have the first codon in the gene codes for the first amino acid. The 10th codon codes for the 10th amino acid. The 20th codon codes for the 20th amino acid. The gene and the protein line up perfectly. Any questions on what I mean by collinear between the gene and the protein? If you know the codon in the gene and you know the amino acid in the protein, there's no scrambling. Any questions on that? Oh, protein. Anybody else? The code is universal. Almost. So I used to stand up and say it's universal until we started sequencing everything. But it's virtually universal. There's one really important exception to universality. So universal means that bacteria use the exact same code dictionary that you do. Absolutely the same. Not a single difference. The exception, the major exceptions, are mitochondria and chloroplasts. So you know that mitochondria and chloroplasts are intracellular organelles, membrane-bound organelles, that have their own genome. And mitochondria and chloroplast genome codes for protein. The mitochondria and chloroplast code is similar, very similar, but not identical. And where it differs from ours is that it's a simplified codon. There are fewer, no, I don't want to go there. It's a simplified codon. It's easier to use. It takes less to decode the mitochondria and chloroplast protein. The mitochondria and chloroplast code. Now, so that's one exception. And everybody knew that. The almost part really refers to the fact that there really are a few very bizarre animals. I don't think they're in plants, but there are very definitely some lower invertebrate organisms that have minor exceptions to the standard code dictionary. And so that's a new wrinkle. Now, if you look back and get some really old stuff, maybe 10, 15, 20 years old, you'll find lots more exceptions. And all of those fell away as people began to do more and more sequencing. They realized that some of the exceptions that they thought weren't real. They were artifacts of editing. So we're not going to really talk about that. But that's a wrinkle in the code. So universal, virtually all bacteria, plants, animals, viruses, everything uses the same dictionary that you have in your book. The mitochondrial chloroplast dictionary is a little bit harder to find. I don't think it's in the book at all. And we're not going to use that at all either. Any question what I mean by universal? The code is ordered. Put a little asterisk by that. We're not going to do ordered code now. We have to go through a lot of stuff before we get to that point. So that's the features of the genetic code. We're going to come back next week and look at order. Any questions on 1 through 6? All right, so that's the features of the genetic code. The next question is, how was the code broken? And so, just like in World War II, there were code breakers. In order to understand that, what we need to do is understand how the code is used. Remember, we're talking about the middle 1950s. In the middle 1950s, nobody knew anything about how proteins were made. That was a complete mystery. Messenger RNA had not yet been discovered in the mid-1950s. tRNAs, an integral part of protein synthesis, had not yet been discovered in the mid-1950s. And so people had to figure out how proteins were made before they could even think about breaking the code. And people knew about ribosomes. People could see ribosomes really are big enough, whether we're talking about eukaryote cells or bacteria. Ribosomes are big enough particles in the cell that people could see them in the microscope. They knew they were there for a long time. As people began to look for how proteins were made, they discovered the function of the ribosome. So ribosomes, in terms of function, 1955. They finally figured out where proteins were made. tRNA wasn't discovered until 1957. It took 2 years after ribosomes were discovered as the seat, as the factory. So ribosomes are where proteins were made, the factory. Transfer RNAs gets its name because tRNAs transfer the amino acids to the ribosome, to the factory, to be incorporated into protein. That came in 1957. Finally, in 1961, mRNA was discovered. The message. Actually, Francis Crick had predicted an intermediate between DNA. People knew that the gene was in the nucleus. They knew that proteins were made in the cytoplasm. Francis Crick predicted an RNA intermediate. But it took until 1961 for people to actually figure out how the code is broken, figure out what the message was, messenger RNA. So finally, in the early 1960s, people could begin to think about how they could break the genetic code, how they could figure out what the codon, the triplet, so codon is 3 nucleotides, what the triplet codons coded for. The way they did that was with cell-free protein synthesis. What that means, the biochemists have been working for 10 years to figure out how proteins were made in the cell. they figured out all the components, the major RNA components, and they were able to duplicate that in a test tube. That's what cell-free protein synthesis means. That's protein synthesis in a test tube, not a cell. And what they decided to do was to use an artificial message. The easiest one for them to make was a message called poly-U. So poly-U is an artificial message made in the lab. Poly-U is equal to that sequence. How many different codons do you see in poly-U? One. What is it?
U.
What's that?
U.
So it doesn't matter where you start. Anybody who knows a little about protein synthesis knows that while I said there are no internal punctuation, we are going to come to external punctuation. And so there is no punctuation at all, internal or external there. There is only one codon. And so the first break in the code came from setting up one, two, three, 20 different test tubes, putting a radioactive amino acid, alanine, phenylalanine, methionine, tryptophan, 20 different radioactive amino acids into 20 different test tubes with all the components, ribosomes, transfer RNA, cytoplasmic enzymes, that clean the cytoplasmic message out so that the background is very low, and added one radioactive amino acid, a different one, to each test tube. And what they found, only one test tube made a protein, and that protein was polyphenylalanine. What's your conclusion from that experiment?
U codes for polyphenyl.
So UUU codes for phenylalanine. Finally, early 1960s, this was a Nobel Prize winning experiment. Finally, the early 1960s, people broke the code, got one codon. If you were in the hunt for the Nobel Prize, what would the second experiment be?
All of them?
What?
The rest of them?
I can't hear you.
The rest of them?
Well, I don't know what you mean by that. Give me an example. What's that? Give me an example. Okay. Poly A. What would you do next?
BBB?
Poly what? T's not an RNA. We already have poly G. Actually, poly G is really difficult to work with because it folds back on itself and messes everything up. What's left? Poly C. So these were the first four.